Thank you for joining me. My name is Peter Nagy. I'm from University of Kentucky. I'm a professor at Plant Pathology Department. Today's presentation is, the title is Virus Replication, the role of the unique pathogenic organelle. Virus is in fact the different kingdoms of life, including people, shown here the COVID-19 causing SARS-CoV-2 virus on the top, and also the mosquito-borne Zika virus, for example, and African swine fever virus, avian influenza virus. These are causing diseases in animals, but plants also getting sick during from viruses, as shown on the right. Most of the viruses are having RNA genomes. These viruses, they don't have DNA, but only the RNA to code for proteins. And these viruses replicate inside the cell, as shown. These viruses induce membranous compartments. These are the virus replication organelles, shown for hepatitis C virus on the left, or poliovirus, and insect viruses like flockhouse virus, and also the plant infecting tomato bushy stunt virus. Today, my presentation will focus on tomato bushy stunt virus, which is a model virus infecting tobacco, tomato, and several other plants. This virus codes just having one single RNA genome, so this is similar to SARS-CoV-2, which also has just one single RNA genome, but TBSV, short for tomato bushy stunt virus, is much smaller than SARS-CoV-2. It only has 5,000 nucleotides. It codes for 5 proteins. P33 is an RNA chaperone, and P92 is the one which is copying the RNA and making a lot of copies of the RNA during infection. And these 2 proteins, P33 and P92, are required for replication of the virus. The other proteins, P41, the code protein, this encapsulates the viral RNA, as shown on the very left of the virions in an electron microscope image. And the other proteins are involved in cell-to-cell movement and responses to the plant RNAi response. We were able to develop yeast as a model host for this plant virus by using plasmid-based expression of proteins and the viral RNA. And this results in replication of the viral RNA inside the yeast cells, as shown now in the boxed area. The very high level of replication takes place. This allows us to use yeast cell biology tools, genetic tools, and also the biochemistry tools. So in plant cells, TBSV induces large replication organelles. The blue on the left shows the viral replication protein P33 inside the cell within the replication organelle. And the green one is a host factor, you can see, and the red one is the viral RNA in the center. These are colocalized, as you can see on the merged image on the very left, very right. And so these are very large organelles. They are about 5 to 10 micrometer in size inside the plant cells. And this is about the size of the nucleus as well. So this is the place where the virus replication takes place. And today I'm going to entertain you about how important is this, the formation of this replication organelle. TBSV forms this replication organelles also in yeast, and this is the tomographic image prepared in collaboration with Cristina Risco in Madrid. And as you can see on the right, the blue ones are the viral replication complexes. These are small spherules, and you see several of them. This is in a peroxisome membrane, which is the yellow one. And then also you can see the gold one is the ER membrane. And so this together, this is the replication organelle. And this is forming during replication. So as I mentioned, yeast allowed us to utilize a lot of tools which is available in yeast to identify host factors, because this virus is like all the RNA viruses of people, infecting people, animals, or plants. They require a lot of resources from the host. And this is what I'm going to talk about regarding TBSV. And to do this, we performed several genomic screens, for example, with using single gene deletion libraries of yeast, and also temperature sensitive libraries of yeast and overexpression libraries. So this covered more than 95% of all the yeast genes, and we identified more than 500 genes which affected TBSV replication. We also perform proteomic screens using, for example, protein arrays, and also we've done mass spec-based after co-purification of the viral replicase complex. We also perform lipidomics to see, because these viruses also depend on lipids inside the cell. So all this allowed us to identify and then characterize several host factors. And today I will show it to you those which we characterized in detail. So the very first thing the virus is doing is that it's pre-assembling a replicase complex. This replicase complex not only contains the P33 and P92 viral proteins, replication proteins, and the viral RNA, which is bound by P33, but it also recruits host factors, host proteins. For example, heat shock protein 70 is such a protein, or elongation factor 1A and 1B gamma, and also helicases. All these are stolen, hijacked from the cell. These are then assembling on the membrane. And this is here shown is that at the early stage, this assembly then also depends on the formation of a special membranous structure. This is so-called membrane contact site. This is where the ER, the endoplasmic reticulum membrane, and the peroxisome membrane comes together based on tethering proteins. These are host proteins also recruited by the virus, and this tethering protein connects these two membranes together, and this allows specific move of lipids from the endoplasmatic reticulum, where they are synthesized to the peroxisome. This allows the virus to create a very special, unique membranous environment, lipid environment, which is critical for the virus to form the viral replicase complexes. This is shown here, and on the very, very right, you can see the electron microscopic image where you see the ER membrane and peroxide membrane, the long lines, they are lining up together, forming these membrane contact sites. Many proteins we identified which are important to participate. These include sterol binding proteins, as well as tethering proteins, which connect these two membranes. They are not fusing, they are just nearby, facilitating the lipid transport. Also, the virus then creates the so-called replication units, replication complexes. These complexes look like here shown, so it's a membrane-based thing. It's almost like a vesicle, but it has an opening towards the cytosol. So the virus has to deform the membrane very heavily to allow it to make a kind of a pocket, and in this pocket the virus replication will take place. The RNA, as you can see here, is synthesized, the viral RNA. At the end after the synthesis, it will be then moved out of the vesicle-like structure into the cytosol, where it will get encapsulated and gets ready for moving to other cells or to other organisms. Here, I'm showing this on the right. You can see now in the box is a spherule forming in a white type yeast cell. You can see that it has a round body, but the opening is the dark one on the top. So this is when it's forming. However, the virus has to recruit membrane shaping, membrane bending proteins. And this is shown here is the escort proteins. These are host proteins, and they are specifically involved in bending the membranes. In addition, the virus also recruit other proteins as shown on the left, and all this is important. As you can see in the next box, one is if one of the escort proteins missing, now then these vesicle structures are open structure, and these are now not supporting efficient replication anymore. Now this is putting it here together, so you can see here that there's on the left a pre-assembly in panel A, and panel B you can see the membrane contact site formation, and that which will make the lipids enriched in a microenvironment for the virus, and then this is followed by bending the membranes into these vesicle-like structures. Here, this was the early stage. However, the replication also creates a peak replication stage where you will see that in addition other now, for example, the early endosome vesicles and the retromer complex also recruited by the virus. And then also you can see now in the next boxed area is that the COP2 vesicles from the ER, which traveling originally to the Golgi, but the virus capturing them and moving them to the replication organelle, which is the yellow bubbles in the middle. So this is now supporting very, very heavy virus replication. One plant cells can support 500,000 to 1 million viral RNA production. This is very heavy factory, viral factory. And now I'm going to show a video. And in this video, what you will see is that how the virus is moving, forcing the cell to move things to the site of replication. The site of replication is, if you look at and to the left bottom, is the purple area, and the green is the actin filaments which the virus hijacks. And so you will see the red small early endosomal structures are moving continuously towards and merging into the virus replication organelle. You see that this is happening in 5 minutes, and all these things will end up in the virus replication organelle, showing how dynamic is this process. The virus is really stealing very important resources from the cell. So again, this is a very, very rapid process and very efficient using the actin filaments. Okay, so until now I showed you that how many protein and lipid resources the virus is taking from the cell to concentrate them to the virus replication organelle. However, also I'm going now to show you that this requires energy, a lot of energy. And what the virus is using, I'm going to show you, is the glycolytic pathway. Glycolysis is using the glucose as a metabolic source, and through a 10 enzymatic reaction, it makes a pyruvate. And during this also ATP is generated. And the advantage of the glycolytic pathway is that it very rapidly generates ATP, and the intermediates of these 10 reactions are important for biomass production like ribonucleotides, lipids, serine, and other amino acids these intermediates are being used. Also that no reactive oxygen production is done during this process. So the virus is hijacking this to the virus replication organelle, as I'm going to show it to you next. So here on the left you see that from glucose to the pyruvate 10 enzymatic reaction, and when we measured the expression of these proteins through their messenger RNA in the infected cells, plant cells, You see the yellow boxes shows that after 4 days, how much more, 4 to 8 times more of these proteins are produced. So the virus is inducing the production of these glycolytic enzymes. And not only inducing their production, but also hijacking them into the replication organelle. So here I showed for example, the aldolase enzyme, FBA1, and you can see that the green FBA1 is now in the merged image, is together with the replication organelle, which is the blue one. And this is only happening when in the virus-infected cells. We've done this with several of the glycolytic enzymes. I'm only going to show you another one, which is a pyruvate kinase. This is the one which is producing ATP. And you can see here is that the green is that the colocalization of the replication organ P33 and pyruvate kinase in cells. So the virus is stealing away these enzymes. And to see that this is important for ATP production, we modified ATP biosensor. This ATP biosensor is based on back in E. coli module, which can bind to the ATP, and during binding, it changes conformation. So 2 fluorophores are connected to this ATP biosensor, and when it bounds to the ATP, then now the 2 fluorophores come very nearby, allowing the so-called FRET, and we can measure the FRET. And we connected this ATP biosensor to our replication protein. So we will measure ATP production around the viral replicase. Here I am showing the results, and so how this works is on the very right you see a scale. The red would mean that lot of ATP is produced, while the cooler colors, including the blue ones, would mean that it's less ATP is produced around the virus replicase. You see that the Y-type yeast, which is on the top, the virus replicase, around the virus replicase, a lot of ATP is being produced. However, when we use a special yeast mutant, which is now temperature sensitive in the pyruvate kinase, so at this temperature now, the yeast is inactivating the pyruvate kinase, and no ATP is produced. And you can see that the virus in this situation cannot have the ATP production. We also done this in plants, and in the top you can see, again, please look at the FRET on the very right. On the top you see in the control that it's greenish reddish color, means that a lot of ATP is being produced within the replication organelle. However, in the bottom you can see when we silenced, we reduced the production of the pyruvate kinase in the plant cells. Now the FRET signal is blue, meaning that the virus now cannot have a lot of ATP at the site of replication. So to summarize this, TBSV is subvert the glycolytic enzymes into the virus replication organelles, VROs. And this is important for local production of ATP, in situ ATP production within the VROs. And so we asked the question, what is the role of this in situ ATP production for the virus? And here I'm going to very much simplify our findings because of time limitations. What we found is that the virus TBSV hijacks helicases from the cell. These helicases are ATP dependent, and they are important in opening RNA structures. These RNA structures are important during replication. And here I'm going to show you the example. So on the left, you see that we have a partial double-stranded RNA, where the top is the black one is the positive strand RNA, and then we hybridized reductively labeled area on the left, which now makes it moving to the top of the J on the right. So the black line, the dsRNA shown there. However, when we have the pyruvate kinase and its substrate, the PEP added to this in vitro reaction, you will see now that the helicases can open now this structure. So you can see the single-stranded so-called ssRNA in the bottom of the gel. So in summary, we found that this locally produced ATP is being utilized by the virus to help the virus replication. So in summary, for this part, TBS induces and hijacks glycolytic enzymes and fermentation enzymes into the replication organelles for de novo ATP production. The advantage is that in this case, the ATP is very rapidly generated within the replication organelles, and there is no competition with the host for the ATP, but first come, first used, so the ATP will be used locally. And also the intermediates of this reaction can be used for biomass production, which the virus will also utilize for its replication. And importantly, also no reactive oxygen is produced during this process. Okay, so this showed that how the virus requires also energy. In the last part of my talk, I'm going to talk about that also the virus requires transportation to make sure that things will be sequestered very quickly to the site of replication to the virus replication organelle. So what we found is that TBSV hijacks the active networks. It modifies it in such a way that it stabilizes the active network. It does not allow its rapid changes during infection. So it basically builds forces the cell to build very active network. And to do this, to demonstrate its importance in what we used in plant is that we took advantage of effector proteins from a bacteria called Legionella. These bacteria produces proteins which can destroy the actin network by cleaving the actin filaments into actin monomers, but those are inactive actin monomers. So here on the top, I show you that the replication organelle, the blue one and the red one together, is, you can see in the merged one, the green is the actin ones. It's matched in the replication organelle. The replication organelle is built, using this actin network. In the very bottom, you now can see the RevK, and RevK, this is the enzyme which breaks down the actin filaments. So you see now in the middle, the GFP mTALIN line, you can see that it's basically no actin filaments are being available in the plant cells. And now the virus replication organelle is not formed. As you can see with the blue and the red is now cannot make this aggregated peroxisome system. And so this is the bottom one. So we also shown that when this happens, when the actin is being destroyed by this effector, then what happens to recruitment of the glycolytic enzyme, the ATP generating glycolytic enzymes? So here you can see that in the blue boxed area is that the ATP generating PGK1 and the virus replication protein within the replication organelles are colocalized. However, when you see the next boxed area, when RevK destroy the actin filaments, now the ATP generating PGK1 and the P33 replication protein do not meet efficiently in the cell, so no signal. You can see also this for the pyruvate kinase now on the right, and when you see the box is that in the control you can see the colocalization, the hijacking of the pyruvate kinase into the virus replication organelle. However, this does not happen when the actin filaments are destroyed. Here this is with the green circle, and So this suggests that the actin filaments are critical for the virus to hijack the glycolytic enzymes. And to show this, that indeed this is also important for the ATP production, here we again use the RefK to destroy the actin filaments. And so circled here with one, you can see that when the RefK destroys the actin filaments, now the replication organelle enlarged here in the boxed area is bluish, which means that the ATP sensor cannot measure good amount of ATP within the replication organelle. This is very different from the top, where you can see that the signal is reddish, meaning that lots of ATP are being produced within the replication organelle. So indeed, the virus requires and depends on hijacking the actin network. So in summary, today I showed that the biogenesis of virus replication organelle is essential for virus replication. So anything where we would inhibit the formation, the biogenesis of this compartment, this pathogenic organelle, then basically we would interfere with virus replication. So the formation of this virus replication organelle depends on the actin network. And also that this allows the virus to subvert the glycolytic enzymes, and this is important for making ATP. And also others, I didn't have time, but also other proteins, host factors have to be subverted by the virus for its replication. And this in situ ATP production allows the virus to use this energy to rapidly replicate the virus in a protected environment. So the take-home message from today is that it takes a village of host factors and hijacked membrane compartments to build the virus replication organelle inside the cell. And again, future research where we would target the formation of this replication organelle would be very likely a very efficient way to fight viral infections. Finally, I would like to say thanks to my laboratory, Judy Pogani and Shifan Zhu, who were running the day-to-day operations in the laboratory. Also, postdocs and graduate students listed here contributed basically everybody to the work presented today. I also thanks the National Science Foundation and National Institute of Health for their continuous support of my work. And with this, I would like to thank you for your attention, and I'm ready to take questions. Thank you very much.